Hi, this is Dr. Smiley and welcome to what I'm going to call the 5-minute focus. We're all super busy, but we can take 5 minutes out of our day to focus on one topic. So let's get started. So today we are going to start a series on macromolecules, and specifically we are going to look at the topic of proteins. So for any macromolecule, we really should be able to name the elements that are involved, the monomers, what it is called when several monomers are put together into a polymer structure, including the bonds that are present, the basic structure that we should look for in that type of macromolecule, and examples and function. So, before we get started, do you think you're able to pause the video and fill any of this in for proteins? So let's get started. The elements present in proteins include carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Recall that carbon makes 4 bonds, hydrogen can only make 1, and oxygen can make 2. Okay, so now that we have our elements, CHON, let's talk about the monomers. The monomers of proteins are called amino acids. Amino acids. Okay, each amino acid is going to have CHO and N present. There are 20 commonly occurring amino acids. The number that are essential depends on the species. Okay, so now we're ready to put our monomers together into polymers. If we put 2 amino acids together, it is called a dipeptide. If we put multiple amino acids together into a chain, this is going to be called a polypeptide. Each monomer, each amino acid, is held together to its neighbor monomer by a covalent bond. Covalent bonds are very strong bonds. In the case of proteins, these covalent bonds get the special name of peptide bonds. So you should associate peptide with proteins. Just with the information that peptide refers to proteins, if I gave you the enzyme called pepsin and said it was produced in the stomach, what do you think this enzyme would break down? Well, again, the same root that we have here, pepsin is an enzyme produced in the stomach that breaks down proteins. Okay, so we've talked about the elements, the monomers, and the polymers. Remember, here the monomers are held together by peptide bonds. Structure. In the structure of an amino acid, we have a central carbon. Recall that carbon is able to make 4 bonds. So each of these lines represents a covalent bond. On one side, we only have a lone hydrogen. On another, we have the functional group COOH, which is the carboxyl group. On the other side, we have the amine group, which you should recognize as NH2. It is worth mentioning that both the amine group and the carboxyl group can ionize in an aqueous solution. So in a water solution, these become charged. So you might see this written as NH3 plus and COO negative. All right, so if we put this aside and get back to our main structure of the amino acid, we have our amine group, our central carbon, our lone hydrogen, our carboxyl group, and then our 4th bond for the carbon is going to be one of 20 different things. This is where you will see the letter R to designate one of 20 different side groups. These are variable. Okay? This is the only spot that the 20 amino acids differ from one another. So to help understand the idea of R groups better, let's look at 2 amino acids. We're going to draw alanine as an R functional group on the left here and serine on the right. So in alanine, the R group is just a simple methyl group, CH3. If you recall, carbon and hydrogen have very equal electronegativity, making alanine a very nonpolar amino acid. In contrast, serine has a carbon that is bound to a hydroxyl group, OH. Oxygen is very electronegative. Therefore, the bond between the oxygen and hydrogen is a polar one, making serine a polar amino acid. One of these amino acids is hydrophilic, that would be serine, and the other is hydrophobic, that is alanine. These properties of different amino acids are going to be important when it comes to how proteins fold. Lastly, let's consider the function of proteins. Because there are so many building blocks that make up proteins, protein function is very diverse. It includes hormones, antibodies, pigments, structural and contractile proteins, transport proteins, and enzymes. We will have to explore this in more detail later because our 5-minute focus time is up. Thanks for tuning in. See you next time. Bye-bye now.